I thank you very much for your very kind introduction. I'm really honored to be the Hitchcock Professor. I wondered why I was chosen to be Hitchcock Professor, and then I received the biography of Lily Hitchcock and discovered that she was interested in volunteer fire companies, and that's the reason I'm here, because I am, in fact, my most important role is as president of the Marlborough, Vermont Volunteer Fire Company. So this may be the first volunteer fireman you ever had talking to you. And I actually go to fires and crawl on my belly and stuff like that. What I want to talk about today is the question of whether we are misled seriously in our understanding of organisms and their development and the mechanics of that development and the evolution of organisms by the kinds of metaphors that biologists use over and over and over again, metaphors that carry with them, as a metaphor must, understandings and meanings which are rubbish in biology, but when you criticize your colleagues for using those metaphors, they say, well, I know that's not true, but I have to explain it to the poor, dumb public, and so on. And the result is that the explanations that are offered, that are current, that are contained in the science section of the New York Times and so on, are just very bad, and they're very wrong, and they give false impressions of what we actually know about biology. So I want to go through some of these rather rapidly and one at some leisure to try to convince you that you should not be misled by what professional biologists tell you. Well, this is a very serious problem. It's the problem of a democratic society is that one assumes that in a democratic society the electorate is educated, yet it is impossible for the electorate to be educated at the present time to an extent sufficient for them to make judgments about the esoteric issues of the elite knowledge of biology or physics. I mean, look, I was a physics student as an undergraduate, but I don't know enough physics anymore to make intelligent judgments about physical issues, and I have to believe what Steve Weinberg tells me. And if he tells me wrong things, I'm stuck with them. So you have to believe what biologists tell you, most of you, because you're not professionals. And it's extremely important that you be cautious about the metaphors that are used. And let me begin with a few and get rid of them rather quickly. One of the most famous pairs of metaphors now in biology are metaphors about DNA. I mean, if you never heard of DNA, you should go home now, because it's not going to be helpful. One metaphor, one statement made about DNA is that DNA is self-replicating. Everybody in this room knows that DNA is self-replicating, right? I mean, you hear it over and over. But that's nonsense. DNA is not self-replicating. It doesn't do any replicating by itself. The DNA is, in fact, copied by a very complex cellular machinery. And notice I use the word machinery, which is already a metaphor, and I'm in some difficulty there. It's copied by a complex cell machinery. It is, in fact, a rather non-reactive molecule, and I want to get to that in a moment. But it does not replicate itself any more than you would say that when you put a letter into the Xerox machine, and make 5 copies of it, that the letter has self-replicated. But that metaphor, the copy of the document in the Xerox machine, is a lot closer to what actually happens in a cell with DNA than the notion of self-replicating. In fact, nothing is self-replicating, no living thing. I mean, except perhaps some organisms in some sense can be said to be self-replicating. I mean, nobody in this room can be self-replicating. You know that. I mean, you might try it, but you won't have much success. So DNA is not self-replicating. It is manufactured by an elaborate proteinaceous and nucleic acid machinery already set up in the cell. That's point one. So never again should look. People sometimes have compared DNA to the Holy Grail. Why? Because the Grail was said to be self-replicating. But the Grail, or rather the contents of the Grail cup, only self-replicated on Good Friday, and whereas DNA is supposed to be self-replicating constantly. But it's a mystical notion that's wrong. Second, DNA is said to make the organism, or at least to make proteins. That's the mantra. DNA self-replicates, and then DNA makes proteins. But that's not true. DNA doesn't make anything. The cellular machinery, again, I have to use a machine metaphor, cellular machinery makes proteins. It doesn't even make the proteins. What happens is the cell machinery uses the sequence of nucleotides in the DNA to create molecules which are strings of amino acids, one after the other in a particular sequence. And then those strings of amino acids are not proteins. They're just strings of amino acids. The proteins are the result of the folding up of that long molecule, a folding which depends completely on the, does not contend completely, but it's wrong to say that that folding is already dictated in the sequence of these amino acids. It depends on the cell machinery, it depends on the milieu of the cell, it depends on other proteins coming along and helping it, chaperoning it, as we say. It depends on the clipping off of bits and pieces. The making of a protein from a string of amino acids is a very complex process which the DNA has nothing to say about. We have an example of that. Those of you who are so unfortunate as to require daily injections of insulin for diabetes know that at the present time it's no longer possible to buy pork or beef insulin. You have to depend on something called humalin, or a similar thing, which is human insulin sequence made in bacteria. The human insulin gene has been implanted into bacteria and they create this thing. The trouble is, when it was first made, the humalin turned out to have the right sequence of amino acids, but it didn't fold up right. And the result was that originally it was physiologically inactive. It was discovered that you had to unfold it again, because the bacteria don't know how to fold human insulin, and refold it under some circumstances which we're not privy to because it's a trade secret. And that made human insulin. And there are problems with that molecule. But the fact of the matter is that DNA does not make proteins. All it does is to contain the information of the sequence of amino acids, and other parts of the DNA have information about when the cell should make it and in what part of the organism. But proteins are the consequence of a very complex process long after DNA leaves the picture. So please, never again believe any biologist who tells you that DNA is a self-replicating molecule like the Holy Grail, and that it makes everything like the Holy Grail. The Holy Grail, by the way, was said to be all nourishing. The Knights of the Grail got all their nourishment from the Holy Grail, which nourished, as it says in the fable of the Holy Grail, sans sergent et sans sénéchal, without attendant and without steward. I mean, it was just there, and it did it all by itself. DNA cannot do anything sans sergent et sans sénéchal, which is the cell of the body. Okay. The third metaphor is that however genes do it, and I want to spend some time on this now, however genes do it, genes determine characters. You have the nose you have because you have those genes. You have the smarts you have because of those genes. I have particular genes which enable me to get up in front of you and speak without being a bit nervous, whereas my musician friends, for example, sweat profusely every time they have to perform in public. They have the wrong genes. Genes make everything. Now, that's also wrong. Genes do not determine the organism. This has to do with the relationship between genes and the way in which organisms develop. And this has to do with the metaphor of development. You may not think of development as a metaphor, but it is. The word develop means literally to unfold, to come out of the envelope. And the notion that organisms develop is inherited from the 19th century. The notion that the program is somehow in there and it unfolds. There was, in the history of biology, a great struggle between two schools, one called the epigenetic school and the other the pre-formationist school. And the pre-formationist school had believed that there was a little man inside each sperm, and that all that happened in the course of the life history of an organism is that little man got bigger and bigger until finally it looked like the men in this room. The egg, by the way, had nothing but nutrient in it. I mean, the real, well, that was that theory of development. And you've seen pictures, some of you, of the little man hunched up inside the sperm. The epigeneticists, on the other hand, say, no, that's silly. There's no little man inside the sperm. The organism actually is created and creates itself out of a pre-existing material, which is quite different from what eventually occurs. And the historians of biology tell us that the epigeneticists won the battle against the preformationists, but they are wrong. The preformationists, in fact, won the battle because I propose to you that there is no difference between the claim that there's a little man inside each sperm and the claim that the complete program will unfold to specify that little man. There's a difference in mechanics, but the notion that all of the information necessary to make each one of you was already contained in the sperm and the egg, of course we've added the egg since then, is a preformationist notion. It is the notion of development, of the working out of the pre-existent program, which is inevitable and unfolds in a succession of stages. But that's not the way life is. Organisms do not develop in that sense. Unfortunately, we don't have another word to use, so I myself will use the word develop. The importance of that word and what it carries is clearer in some other languages. I mean, for example, in Spanish, the word for development is desarrollo, the unrolling. In German, it's Entwicklung. Entwicklung means I take a ball of yarn and I throw it out and it unrolls. It's an unrolling. And that's the German word for biological development, Entwicklung. And I once asked an Israeli friend of mine, I want to take advantage of a historical experiment. Hebrew, which is now spoken, being a biblical language, had no words for development of an organism. So they had to make them up, like they had to make up words for railroad and telephone and so on. And I asked him, what word did you invent to stand for the development of an organism? And he told me, and I think I got this right, if there are any Hebrew scholars here, forgive me, hitpateach, which is the same word in Hebrew as is used for the developing of a film, which has, of course, the image latent in it already, except it's in the reflexive form. So an organism develops itself just as a film is developed in the developing bath. Organisms don't develop in this way because it is not the case that all the information necessary to specify an organism is contained already in the fertilized egg. It's just not true. And I want to show you a few examples of this, but I want to quote one of the most eminent biologists, a really eminent molecular biologist of the present time, Sid Brenner, who gave the opening address at the 100th anniversary of the publication of Darwin's opus. No, I'm sorry. It was the 100th anniversary of the death of Darwin, which is perhaps more to the point. And what Sid said was, and I heard him say it, if you gave me the complete DNA sequence of an organism and a large enough computer, I could compute the organism. Now I have to say, although I respect Sid as a very great scientist, that statement is rubbish. It is not true that if you had only the complete DNA sequence of the organism and a sufficiently complex interpreter, that the organism would be developed. Because the organism does not compute itself from its DNA. There is other information, important other information, in producing an organism. And Ellen, are you here, Ellen? Yeah. Ellen Gobler is my visual aid. And I want to show you an experiment done in California which illustrates this as no other experiment has ever done. This is a cloning experiment. This cloning experiment was done in 1938. I mean, cloning is nothing new. Plants are easily cloned by simply taking scissors and cutting them into pieces and planting them. And those pieces planted from that cut-up plant are genetically identical. And you can then grow them in different environments and see what you get. This is the famous work of Clausen, Keck, and Heisey, done at the Carnegie Institution of Washington Experiment Station in the Sierras. And what you see across the bottom, this is what Clausen, Keck, and Heisey did was to take sample plants from nature, cut the plants into 3 pieces, and plant one piece at a low elevation, one piece at a medium elevation, and one piece at a high elevation. So these are the 3 pieces, this one, and this one, of the same genetically identical plant. These are 3 pieces of the same plant. These are different plants. So the different genetic types are here. The clones of each genetic type are vertical. Is that clear to everyone? And look what happens. The plant that grew tallest at the low elevation did the worst at the medium elevation, but in fact it did the best at the high elevation. The plant that was second tallest at the low elevation was third, fourth tallest at the medium elevation, and sort of in the middle here, and so on. You notice they all flowered at the highest elevation. Some of them failed to flower at the medium elevation. And there's no relationship between the growth of the plants at the low elevation and how the very same genotypes grew at the medium elevation and the high elevation. If you actually measure the heights in plants in centimeters, you'll find a 0 correlation across environments. Now, Dr. Brenner, how is it possible to make the statement that the organism can be computed from its DNA when the organism has failed to compute itself from its DNA? Which of these sets of DNA, which of these DNA samples gives you the best growth? And you can't answer that question until you also say in what environment they were grown, because the order has completely changed. And this is a classic experiment which is seen over and over and over again under different circumstances when this kind of experiment is done. That at minimum, you must not only say what the genetic differences are, but you must also say what environment those genetic differences were manifest in. Development occurs within some temporal sequence of environments, and there's a lot of information in the environment as well as in the DNA. This is the most striking example, and people say, well, I know that. I mean, everybody knows that. Everybody knows that there's got to be some effective environment. After all, if you have the genes that would make you fat and we starved you, you of course would become very thin and finally die of malnutrition. So there's some kind of general effective environment. And that is illustrated in a picture I drew freehand from a picture in Arthur Jensen's famous monograph about IQ in humans. And what Arthur Jensen was illustrating for him was the effective environment on IQ. The different lines here are different genetic types, and we won't go into the issue of who he thinks belongs to this genetic type and that one. The important point for him was that there are some genetic types that have low IQ and some high. Of course, if you put them all in a deprived environment, there wouldn't be very much difference between them. And if you put them all in a very enriched environment, there would be more difference. But the important point for him was that the one that was lowest in the deprived environment is also lowest in the enriched environment. There's just so much you can do with environment. Now, this is a very different picture from the one I just showed you. And there is no evidence whatsoever that genotypes are like this. That's just made up. It's made up in order to agree, yes, environment matters, but the order of the genotypes is what counts. And therefore, if you're smart in a deprived environment, you'll be even smarter in an enriched environment. So we don't have to talk about environment anymore. It is sometimes said, well, that may be true for those plants that you showed us, but it's not true for animals, it's not true for well-known mutations and so on. I want to show you, Ellen is going to help me by showing you a couple of other pictures, just to give you an impression of the broadness of this phenomenon. These are pictures of the number of abdominal bristles on the belly of fruit flies. Okay, just to be as different as I can from plants, and there are 3 different temperatures at which the fruit flies developed, and that line is the number of bristles at low temperature, medium temperature, and high temperature for one genetic type, this one for a completely different genetic type, this one for a complete... There's no order to it. Everything gets completely confused. Some get more bristles as it gets colder, hotter, some get more in the intermediate and then come up again. There's no pattern. Okay, so that's abdominal bristles in Drosophila. Another case is corn. Everybody has heard about how modern corn gives you a higher yield than old-fashioned varieties of corn. This is a comparison of an old variety of corn, no longer planted, this one here, and a new variety, this one here, which does better, gives you a higher yield of corn in all environments until you get to an extremely good environment, and then the old variety does better than the new one. That is to say that these cross each other. And the fact of the matter is that most farmers are growing corn in these environments, so they prefer this variety, and I understand that. But it is not the genetic and developmental case that this type is unambiguously better than this one. It depends on the environment. And finally, one more example, and then I won't bore you with this particular point anymore. My geneticist friends say, well, that might be true, but what about famous mutations in Drosophila? Mutations that really interfere with development. Well, here's a famous one. Drosophila have compound eyes. They're sort of oval. And one mutation will reduce the size of these eyes to a bar-like appearance. And here is how normal, so-called wild-type flies, the size of their eyes as a function of temperature, and as it gets warmer, the size of the eye gets a little smaller. Here is a mutation called infrabar, which reduces below wild type, or below normal, the size of the eye, but in this case it increases with temperature instead of decreasing. And here's another mutation, related, in which it decreases with temperature. So both of these are unambiguously smaller than wild type, but they have completely opposite environmental effects on their development, and they cross each other. So that's an example of the most extreme kind of environment. Okay, so that's the first point I want to make, that you at minimum have to specify the genotype and the environment, developmental environment of an organism before you can specify what the organism looks like. So we have different models. The top model is the one where what matters is genes, and all environment does is to put in various factors. There's a genetic plan A and a genetic plan B, so the organism A comes out one way and the organism B comes out the other, and the environmental factors are simply the sort of nutrient material necessary for development. Another plan is to say environment counts for everything and genotype counts for nothing. And there are such characters. The phonemic structure of my speech, for example, is, as far as I know, undetermined in any way by my genes. But the fact that I can speak depends on my having the right genes, because no monkey will ever get up and give the Hitchcock lectures, because those monkeys don't have the right genes, not because they don't have the right environment. So different environments will give rise to different organisms. Organism A has the phonemic speech of an educated American. Environment B will have the speech of a Chinese, for example. But we all have to have the general genetic rules of tongue and voice box and brain and so on. That's the alternative, the opposite alternative. And then there's the possibility of what I've just been talking about, developmental interactions between organism and environment, in which genes matter and environment matters, but there's no simple ordering of the effect. And you get different organisms depending both on the genotype and the environment in no particular order. Now, what I want to say is that this scheme is still insufficient to understand organisms. There's yet another factor in what we call the unfolding or development of an organism, which is neither environment nor genotype. If when you go home you will please hold up your fingers to the mirror on your right and left hand, you will, most of you, discover that there seems to be no similarity, or rather little similarity, between the fingerprints on your right hand and the fingerprints on your left hand. I've actually seen the fingerprints of Galton, one of the most famous 19th century heredity researchers, and you'd never know those fingerprints on the left hand came from the same person as on the right hand. And that's well known among fingerprint experts. But look, the fingerprints, which are the folded ridges of your skin, developed inside your mother's womb while you were a fetus. The genes on the right-hand side are the same as the genes on the left-hand side. There are occasional mutations in the cells, but in general they're the same. And the environment on the left and right-hand sides inside the amniotic sac would hardly be called different on the left and right-hand sides, yet you have completely different fingerprints on the two sides. Indeed, all symmetric, so-called bilaterally symmetrical organisms are in fact asymmetrical. Fruit flies have different numbers of bristles on the right side and the left side, not on the average, but one will have 7 here and 5 here, another will have 6 here and 4 here, and so on. On the average, it averages out. People are asymmetrical in many, many respects, including their fingerprints. What is the origin of the asymmetry of development, on the fluctuating asymmetry of development on the two sides of an organism when the genes are the same and the environment is the same? You can't talk about a different environment on the left and right hand of a fruit fly, which is in development, which is no bigger than the end of a pencil suspended on the inside of a milk bottle. I mean, it doesn't make sense. And we know the answer to that. The answer is that there's something which is sometimes called developmental noise, which doesn't say anything, which means that the actual division of cells, the timing of division of cells, and the exact ordering of those divisions in a line and so on depends upon the molecular state of the cells, internal to the cells. But the number of molecules inside any cell of a particular kind is very small. It's not like the chemists who tell us there are Avogadro's number of molecules in a test tube. There are 7 of these and 3 of these and 9 of those. They're the essential molecules that are important in the division of the cell. They're disposed around the cell in different places. They have to interact with each other, and it takes time for them to come together. They have to be in the right vibrational state, and this is real quantum uncertainty. They have to be in the right vibrational state when they meet each other. And all of that is subject to a kind of indeterminacy from the quantum level up to higher levels of indeterminacy. And that means that whether a cell divides at a particular moment and how often the progeny cells divide in a given period of time has a random component to it. And I could tell one of these random stories at length, but I won't. This has a profound effect on what happens. Let me give you another example and then not go on with that. If you take a single bacterial cell and you sow it in a huge flask of culture medium, which is constantly being stirred, so there's no chance for the medium to separate out in any way, that bacterial cell will divide in about 63 minutes if it's E. coli at the right temperature. The 2 cells that have come from that cell then divide, but they don't divide simultaneously. First one divides, and then a few minutes later the other one divides. Those 4 cells don't divide simultaneously. First one, then the other, then the other, then the other. That's also true in the development of embryos. The 1-cell stage gives rise to the 2-cell stage. In your textbooks, you're told the 2-cell stage then gives rise to the 4-cell stage, but that's not true. What actually happens is, after the 2-cell stage, there's a 3-cell stage, and then a 4-cell stage. There's asymmetry from cell to cell, and lack of synchrony, and pretty soon in a bacterial culture, a cell is dividing every instant, but it's some other cell. And the reason is, and we have quite good knowledge about that, because if some molecule has to be in a certain concentration in the cell for division, say 7 copies, after the division, there'll be 4 copies in one cell and 3 in the other, or worse still, 5 in one and 2 in the other. Well, it takes a different amount of time for the cell to manufacture the missing copies, depending on whether you're starting with 4 or 3 or 2. And that results in a time lag, and those time lags can have profound effects on major external appearance, because timing of development, movement of cells from one layer to another, and so on, have a big effect. Some things will fail to develop if the tissue doesn't divide it in the right moment, and so on. So these quantum and thermal statistical effects have a profound effect on development. And so we do not know what an organism will look like, even if we know the complete genotype and the complete environment of the organism, because there is that random uncertainty. And that's summarized in the next slide. These are girls who some of you may know. They are the Dionne quintuplets. When I was a boy, I couldn't go to the movies without being treated the Dionne quintuplets. For those of you who have been born too late for the Dionne quintuplets, these were identical quintuplets born to a fairly poor family in rural Ontario. They were taken from birth by the physician who delivered them and the province of Ontario and put into a zoo where you could see them being taught, talking, playing. You notice they are identically dressed, identically shod, identically coiffed. Everything was done to make them identical. First of all, their genes were identical. And secondly, their environments were made as identical as possible by a deliberate public relations gimmick. And these girls then grew up and became very angry at the way they were treated. But the important point is that the life histories of these girls, despite their genetic and environmental identities, grew up to have very different life history patterns. Two took on a religious vocation, 3 were married, a couple had children, one died of a mental in a mental disorder. We don't know what she actually died of. Another one committed suicide, and only 3 of them are left. I was going to show you a picture of the 3 surviving girls, now women of 60. They look sort of like sisters. They do look like sisters. They look rather similar, but their life history patterns were utterly different. So, even though every attempt was made to make them identical, it failed. And that is a consequence of that developmental noise. Social developmental noise. Developmental noise in the development of their nervous systems. So the 2 wanted to be nuns and 3 wanted to be married women. I say to you then that if you want to understand an organism, you have to know not only about its genes, not only about the environment, but you must have adequate models of the randomness of development, both mental and physical, in order to have a complete explication of the organism. So the metaphor of determination is not a correct metaphor for anything biological. Biological organisms are not determined. They have a degree of indeterminacy which is as important as the quantum indeterminacy of elementary particles. Now I want to spend the rest of my talk on yet a last metaphor, which comes from evolutionary biology. And that metaphor is the metaphor of adaptation. According to the vulgar theory of evolution, organisms evolve because genetic variation occurs, that's certainly true, and some individuals leave more, some kinds leave more offspring than others, that's natural selection, that's certainly true. The consequence of that variation and differential reproduction is that the organisms become adapted to the environment around them. They start out not being adapted, they start out, in Darwin's terms, not fitting into the hole in the environment which they find, but they get changed in their shape and morphology and behavior until they fit better. And the notion of fit and fitness, which is a Darwinian notion, was literally meaning fitting into a hole. And the notion of adaptation then is the notion that there exists out there in the physical world, outside the organism, a place, a kind of ecological place for that organism. And the evolution of organisms is the shaping of the organism to fit into that hole and the mechanism of natural selection. Now what I want to say to you is that adaptation is the wrong view of evolution. Organisms and their environments do indeed seem to be marvelously fitting, but that is not because organisms adapt to a pre-existing environment. For organisms to adapt means that the whole has to already exist before the organisms do. When I go to Europe, I take an adapter for my electrical stuff so that I can plug it into 220 volts. That is, adaptation implies a model to which you move, and that's what the word means, adapt, become apt to. But the fact of the matter is that the environments of organisms do not exist before the organisms. The physical world exists, but the physical world is not the environment of any organism. The environment of an organism is a juxtaposition of bits and pieces of that physical and external biological world, which juxtaposition is created by the sensuous life activities of the organism. Imagine describing the environment of an organism that you've never seen. There's a non-countable infinity of ways that you could put together the outside world, but almost none of them are occupied by organisms. You have to take the word environment literally, environ, around. The environment is organized around the existing organism. And there are no environment, just as there's no organism without an environment, there is no environment without an organism. We have a famous experimental evidence of that, and that was in the design of the Mars lander. There were two competing designs for the Mars lander. One was, I think, Josh Lederberg's, which was essentially to send up an electron microscope and a long sticky tongue, and the tongue would unroll on Mars and roll back, and there would be dust on it, and the electron microscope would look at the dust, and if you saw anything that wiggled, you'd know there was life on Mars. Or at least if it didn't wiggle, it had the right shape. That's the morphological definition of life, and I don't know why some people are laughing. Why are you laughing at the morphological definition of life? What you should laugh at is the way they decided to do it. Being scientists and much more sophisticated than poor Josh Lederberg, they decided you can't tell life when you see it, so you'll determine it by its physiology. So what they did is to send up not this sticky tongue and vacuum and microscope. They sent up a vacuum cleaner, a tank-type vacuum cleaner, with a hose that went out and sucked up dust. The dust went into the vacuum cleaner, which was filled with a radioactively marked substrate, sugar, so to speak. And if there was any life on Mars, it would break down the sugar. The carbon dioxide, which was released, would then be radioactive carbon dioxide, and it would be detected by a radioactive detector in the machine. And sure enough, when the lander landed and sucked up that dust, the number of counts per minute of radioactive carbon started to go up and up and up and up and up. And I mean, I wish I'd been there. People must have been jumping up and down with mad glee. And then suddenly it shut down, just bang. And everybody assumed that the machinery had shut down, like this machine here. But it hadn't. All the tests showed the machinery was still working. So there had to be something else that had happened. Well, there was a meeting at MIT to discuss this problem. I remember the meeting. And to make a little fun of it, what they finally did was to take a vote. And the vote was there was no life on Mars. I mean, that's not literally what happened, but that was the outcome of the... And what was decided instead, correctly, was that what had been observed was the breakdown of the organic material and the evolution of carbon dioxide by a catalytic reaction that occurred on the finely divided clay particles or soil particles from Mars. And that was reproduced on Earth later in the laboratory. You could reproduce that with finely divided clay particles. Now, that's the one you should laugh at, because the naivete of the scientists, very good scientists, it's easy to call them naive at a distance, that assumed that you knew what life on Mars ate. That is to say, you knew what the ecological niche of life on Mars was, so you provided an ecological niche, and if there was life on Mars, they would live in it. But how can you know what the ecological niche of life on Mars is? Indeed, there are a lot of organisms on Earth who wouldn't ferment that broth. I mean, there are sulfur-fixing bacteria and all kinds of anaerobic organisms and so on. Why assume that the life on Mars is Escherichia coli, or whatever it is that they thought it was? This is a demonstration of the fact that you cannot specify the environment of an organism that you have never seen. The environment of an organism has a relationship to the organism which is complex and has to be understood in its entirety. And I want to go very briefly, because my time is fleeing through the elements of that process. What I want to say is that organisms construct their environments. They don't adapt to them. It's not that the environment is fixed and the organism moves to it. The organism evolves, and in evolving, causes the formation around it of an environment which itself is also evolving. Organisms and environments co-evolve. And that co-evolution of organisms and environments has to be understood. And I think the proper metaphor, if there is one, is the metaphor of construction. First of all, organisms decide, and let me use the word decide, but you'll understand I don't mean that in a vulgar sense. Organisms decide what aspects of the external world are relevant to them. When I lived in Britain, there were birds, thrushes, that broke snails on the roof tiles of my house to eat the inside of the snails. But there were also great tits and ravens and so on who didn't do that and didn't come anywhere near the house. Now those roof tiles were a very important part of the environment of the thrushes who used them to make food, but they were not part of the environment of the crows. The fact that that a stone or a roof tile is part of the environment of a bird depends on whether the bird, in a sense, interacts with it in a meaningful way for its life. The fact that it flies over it doesn't make it part of its environment. Propinquity is not what determines whether whether something is part of your environment. What determines it is the functional interaction you have with that bitter piece of the world. It isn't just that birds make nests and moles burrow and people build beautiful buildings. Every organism is in the process of constructing an environment in the sense of making some things relevant and other things irrelevant by its behavior, its morphology, and its physiology. Everyone in this room at this instant is creating a personal environment. That environment, if I could take pictures of you, and I've seen these pictures, so this is of you using what I call Schlieren optics, which depend on the different optical density of refractive index, actually, of a medium through which the light is passing. When I take pictures of you, what I would see for each one of you is that around your body is a layer of moist, warm air, which is rising continuously and going off the top of your head. That moist, warm air is being created by your metabolic activities. You're creating the heat and you're creating the moisture. You live not in the atmosphere of Berkeley. You live in the boundary layer which you are carrying around with you and creating the moist, warm air. And the little buggies that may live on you are living in that boundary layer. And if they get, for example, too big, they'll stick out of the boundary layer and they'll be in a different world. The evidence for that boundary layer and the importance of it is what the newspapers report as the wind chill factor. Have you ever thought of why it gets colder when the wind blows? Why should it get colder? I mean, you get more molecular movement, you've got to get warmer. It's not because the wind is colder, it's because it blows away, not only from your individual bodies, but from the buildings in which you are, from every part of the world in which you're living, that boundary layer, and exposes you for the first time to the world out there, the real world out there. So that's an example of construction. And construction goes on at all times. So organisms determine what aspect of the physical world is relevant to them, and they construct out of those relevant bits and pieces a world of interactions. The third thing that organisms do is that they change that world in which they're interacting. They both create it and destroy it. Every act of consumption is an act of production, and every act of production is an act of consumption. By the way, a prize will be given to any of you who know the origin of that quotation. Grundrisse, in fact, yes. Good on you. You get the prize. Every organism is in the process of using up the materials it needs and for its life activities and depositing in the world waste products. Not just humans. I mean, every worm, every bird, every bacterium is destroying the world in which it lives at the same time that it's consuming it. Moreover, organisms are in the process of producing the world in which they live. It isn't only humans that farm. Fruit flies farm. You can do experiments to show that if you put a fruit fly in a little bit of medium and put some yeast on top for the fruit flies to eat, those fruit flies survive better, the larvae, survive better if there are more larvae than if there are fewer, which is not what we usually think, because the more there are, the more competition. But in fact, what happens is that the larvae burrow in the medium and create tunnels, and in the tunnels the yeast grows, and therefore there's more food for them. Now, if you put enough, of course, then the consumption exceeds the production. But every organism is both producing and consuming. And many organisms are in the process of producing their own life materials. Plants do that all the time. Plants change the physical nature of the soil in which their roots grow. They break it up and change it. They exude chemicals called humic acids through the roots. Those humic acids encourage the growth of fungi called mycorrhizae, which are important in the nutrition of the plant. So the plant is farming fungus, which is then nourishing the plant. And every organism is doing that at all times. It is reducing and increasing the materials of its life. So organisms both create and destroy. Mort Saul, some of you may know Mort Saul, he once said, no matter how cruel and nasty and vicious you have been today, remember that every time you take a breath, you make a flower happy. So the fouling of your own world may be the enrichment of someone else's. Putting aside the joke, there are features of the occupation of the earth which are very clearly examples of that. I come from New England. I live in a small town in New England, and there used to be a lot of pine forests there. There were so many pine forests that wood companies and paper companies came in and cut them all down, bought them first of all, and then cut all the stuff down, figuring when the new ones came up, they'd have a perpetual source of softwood for paper and lumber. But when they cut the pine forest down, instead of pine trees coming up came hardwoods, which were totally useless to them. And it was discovered by a long series of historical observations and experiments that the history of New England pine forests is that farms, when abandoned, as many farms were after 1840, the maximum population of my town was in 1840, and then decreased as every sensible person quit the stony soil and went west to Pennsylvania, Ohio, and so on for the deep soils. When farms are abandoned, the first thing that comes in is herbaceous vegetation, and then pine trees come in, blown in from other pine stands. Those pines grow up, but those pines produce shade, which makes it impossible for their own offspring to grow. So pines have invented the conflict between generations, so to speak. They create a world which makes it impossible to replace themselves, and instead of that, hardwoods come in. This is known as the white pine succession, and that's very common. And pines are what we call weeds. Weeds are not the things that grow in your garden, necessarily. Weeds are a kind of life form which depend for their growth on disturbed habitats. They come into disturbed habitats. They then change the habitat in such a way that another generation of the same species cannot grow there. And so when they die out, yet other species come in. They depend on disturbance, and then they change that world to be inimical to their own kind. And that's a very common form of life among weeds. Finally, well, two things. Another thing that happens is that organisms change transduce the physical signals that come into them. Your livers are not getting warmer as a consequence of the hot air that I'm producing, because the impinging thermal activity of molecules is converted by your physiology, which is in your genes, of course, into changes in the concentration of various hormones and other substances in order to control your body temperature. That happens, the one I think of most graphically is what happens to me when my wife and I go out collecting fruit flies in the California desert in Death Valley. Once in a while you see a sidewinder. I don't know how many of you ever come across a sidewinder, but when I come across a sidewinder, the photons that have impinged on my retina and the rarefactions of air that have impinged on my eardrums become transduced immediately into a form of chemical information, namely my adrenaline goes up. And I assume that that is specific to my genes because I'm sure that when one sidewinder sees another one, its adrenaline doesn't go up. Maybe some other hormone goes up depending on the sex of the sidewinders. But the transduction of external information is a function of your particular biology, and it's not automatic in the world. So the environment that you perceive is different from the physical nature of what comes in on you. Finally, and that's the last thing I'll say about this, organisms are mathematicians and statisticians. Organisms carry out both mathematical integration and mathematical differentiation. The integration is time integrals. Organisms time average over long periods of time. Trees flower when a sufficient number of either degree days or light days they accumulate the information until it reaches a critical level, and then they flower. They lose their leaves when both degree days and light days have reached critical values. They're averaging all the time. And one way to average is to store. Energy storage is a form of time averaging. Acorns store the energy of photosynthesis that has been put in. No plant dies at night just because it can't do photosynthesis. And plants and trees don't die in the winter because they're not photosynthesizing, because they have methods of storage. The potato is the storage of a potato plant. Then we take that storage and we store it, so that the price of food does not fluctuate immensely from summer to winter. It's averaged out because we have mechanisms of storing energy. And then we have personal mechanisms of storing of energy, which is the greater omentum in men and other parts in women, which are storage mechanisms. Then we have social storage mechanisms. The potlatch of the Northwest Indians is a method of storing of goods over time. And of course, money is a form of storage. Futures, the futures market irons out the cost of food over long periods of time. It's kind of information storage. And organisms differentiate. They see, for example, looking. When you see something, it requires a kind of contrast between light and dark. You see edges, and if that contrast is not proper, you can't see the transition. But the most famous example of doing differentiating, of detecting rates of change, is copepods, little tiny water fleas, worked on by Banta back in the 20s and 30s. What Banta found, water fleas have both sexual and asexual reproduction. Usually they don't produce any males, they produce asexually, producing asexual eggs. But once in a while, males will suddenly be produced, and they'll mate and they produce sexually. Why? It turns out that they are sensitive to the rate of change of external cues. If the temperature suddenly shifts upward or downward, they will produce males. If the amount of oxygen in the water goes up or down, they will produce males. If the amount of food in the water increases drastically or decreases, they produce males. Males are produced as a consequence of rapid changes in the environment, not as a consequence of the average level of the environment. Let's say those organisms are measuring time rates of change, and their physiology is sensitive to time rates of change. So to put it all together then, I want to say to you that you should not think of evolution as some fixed set of problems to which organisms adapt. The reason organisms seem so well and wonderfully fitted to the world in which they live is because they have, in large part, not entirely, accumulated a world around them which is appropriate to their own morphologies, physiologies, and metabolisms. Of course, they also change, and as they change, their environments change, and environments and organisms are co-evolving at all times. And that's why they seem indeed so well adapted to each other, why the fit seems so good. So when you think about the evolution of life, you mustn't think about it as the movement in a fixed world. And that means, finally, let me say, that has some political implications. The slogan preserve the environment is a slogan that makes no sense in biology. What you have to say is that every organism has a different environment, and those environments are changing with the organisms, and all environments are at all times in the process of change. The political issue then is not stop the world, make the environment, which doesn't exist, or even the human environment, which doesn't exist, the same as it used to be or is now. The political issue is to decide what kind of world you want to live in, and then do your best to direct the change in the inevitable change in environment, which living and metabolizing creates, in some directions which will be as suitable as they possibly can for human life and perhaps for animal life and plant life. That's an issue which is not a scientific issue. But it must be done on the basis of knowledge of how environments are changing and an attempt to funnel those changes. You must stop forever this constant repetition of let's preserve the environment. And by the way, you can't even talk about preserving species. You can talk about reducing the rate of their extinction, but you can't stop it. 99.9% of all species that ever existed are already extinct, and there's only there are two laws of life. One is that all flesh is mortal, and the other is that all species will become extinct eventually. I mean, if not tomorrow, then when the sun becomes a red giant and burns up the earth. So the issue is to direct the direction and the rates of change, not to prevent change. Organisms are by their nature evolving, and the environment in which they live is by their nature evolving. So please do not become victims of these metaphors. Let me leave you with, I don't know what to call it, a bon mot of Norbert Wiener, who in a paper which is not generally known very well on science and its forms, said that you cannot avoid metaphors. I couldn't avoid them. I talked about the machine metaphor and so on. Since we have to use metaphors, we have to be careful. And what Norbert Wiener said is, the price of metaphor is eternal vigilance.